So Marom is Cattell Professor of Biomedical Engineering at the City College of New York and the City University of New York, and is co-director of the Neuroengineering Group and the New York Center for Biomedical Engineering, and is also co-founder of the Soterix Medical Incorporated. His technologies and technologies developed by his group have been used in clinical trials in over 100 medical centers. He's published over 200 papers and book chapters and is inventor on over 30 patent applications. He co-invented high-definition transcranial direct current stimulation, a non-invasive targeted and low-intensity neuromodulation technology. And I should tell you, too, that Marom is a good friend, and I feel really happy that he could make it today. He's a great engineer and really helping to bring tDCS and electrical stimulation along almost into the 21st century, when honestly in my lab we still use equipment that looks 100 years old, but we could do better, and Marom's really helping. So please join me in welcoming Marom Bikson.
Thanks, Vince, for that introduction. Good morning, everyone. Let me jump right into it. But definitely want to thank Vince for organizing this amazing meeting that is integrating imaging and stimulation, which is something that really Vince has already been doing for a very long time. But I think, in a sense, the field is sort of catching up to where he's been for quite some time. I'm going to be talking about transcranial electrical stimulation, and particularly tDCS, and how to combine that with EEG. And in the context of the EEG work, a lot of that work was done by Franca, who's here, and her group in Italy. These are my disclosures and my support. So I'm not going to spend too much time introducing transcranial direct current stimulation, tDCS. I'm assuming most people in the room have heard about it in scientific publications, in the press. It's apparently a very simple technique that uses low-intensity direct current stimulation applied non-invasively to electrodes on the scalp. Some of that current reaches the brain, and it's speculated to be able to change behavior, learning. It's being investigated for a very broad range of neuropsychiatric diseases. There's been an explosion of interest in it since seminal work from Michael Nietzsche, who you'll be hearing about, and we'll probably be giving a much more comprehensive introduction to tDCS mechanisms. These are sort of a informal list of the kind of things people have been suggesting tDCS works for. Obviously, pain itself is many things. Depression is many things. If you get to the end of that list, you start to see things that are more creative. And so wherever you stand on your enthusiasm for tDCS, the sheer breadth of the applications that it's being investigated for is pretty striking. And I'm not going to get into any particular indication per se, but I think no matter where you stand on sort of the spectrum of skepticism, and belief, clearly with so many indications, we need to be thinking about how we're customizing tDCS, how we're optimizing it for any given indication, and that's really what I'll be speaking about today. So with conventional tDCS that uses two relatively large electrodes placed across the head, these will produce current flow in the head. I'll be showing you images like these. These are false color maps of computer simulations. Generally, when you place an electrode on the head, there will be a bit of current in the cortical region underneath that electrode, but that current won't just disappear when it reaches that part of the cortex. It has to keep flowing until it gets to the other electrode. And as a result, you're always seeing two electrode effects, and as a result with tDCS with two electrodes, you're typically also seeing deep brain regions that are affected. So it doesn't mean that you cannot optimize this approach, but there are certain limitations when using two large electrodes. Now, using an extracephalic electrode does not negate this problem. It's still the case that you can often concentrate electrode under a current under one electrode in that cortical region of interest, but it has to get to the other part of the brain. And so if we look at the brain from the bottom, you can see all that current exiting as well. So again, using two large electrodes, you have to be thinking about current entering and exiting and affecting all the brain regions in between. There are other ways to do tDCS, and this is the 4x1 montage of high definition tDCS. In this case, you have one center electrode that's sort of your active electrode of interest, and you surround it with four other electrodes, and these act as the returns, and they'll have the opposite polarity of the center electrode. And this montage, this approach, is presumed to produce much more targeted brain current flow. Essentially, the tighter you make the ring, the more focused the stimulation will be. And it's also largely restricted to the cortex. And this was something we first introduced, I guess, over half a decade ago. Another nice feature about the 4x1 montage is we believe that it's largely unidirectional. You have one, let's say, center electrode. Let's say that's the anode on the right. That makes the outer four cathodes. They will have a quarter of the current of the center electrode, and on top of that, that current will sort of be returning through much of the brain. And so the result is when you do 4x1, you can produce largely unidirectional neuromodulation. We speculate compared to conventional tDCS where you have two electrodes. And so this image illustrates the only thing different between these two images is one is center anode, one is center cathode. And in this case, what the picture is showing you is where current is flowing in red and where current is flowing out in blue. This was a long process to develop this technology. It did start with computational models, but we did need to advance the state of the art of the models to do it. Really, starting around 2005, 2006, we developed the first gyri-specific models of the head, of the entire head. These did not exist in a precise form until then, and we needed these tools in order to develop these new montages. And then all the way back in 2008 is when we presented the use of high definition, and particularly the 4x1 montage. And again, we were starting with theory. So from 2008 to 2009, those papers were appearing. Around the same time, we had the first tolerability experiments with Eric Wasserman at NIH, followed by pilot experiments looking at its effect on both experimental and real pain. And it wasn't until 2 years ago where Michael Nietzsche, who played such a seminal role in tDCS, demonstrated that high-definition tDCS produced equal or greater changes in neuroplasticity. And additional validation has followed, but now it's certainly use is certainly expanding. But it was a long road, and there were really two reasons. One reason people thought it wouldn't work is they said, well, the skull is resistive. People were very familiar with transcranial magnetic stimulation. The skull is transparent to magnetic fields, and therefore with TMS you can be as focal as you want. The skull is resistive to electricity, and therefore don't bother to even try to be focal with transcranial electrical stimulation. But what we discovered is actually, sort of ironically, the skull is the one place where current does not disperse. It actually goes straight through because it's highly resistive and it doesn't want to move around. It disperses in the CSF and in the skin, and that can be partially controlled by putting a ring around your active electrode. And prior efforts, including foundational efforts in tDCS, weren't optimized for focality simply because they were using large pads. It wasn't the physics that were limiting things. The other thing that people thought would not be possible was that you couldn't pass direct current through a small electrode. So people were used to using large sponges. 2 milliamp was the highest intensity people were using for about 20 minutes. And the feeling was we're using these large sponges because if we went smaller, we would produce skin irritation and skin burns. And it's true that if you take a sponge and you make it smaller and smaller and smaller, eventually you get to the point where you will produce skin irritation with 2 milliamp in 20 minutes. But a sponge is hardly a state-of-the-art technology. And so we spent what really was about 3 years using modeling and iterative approaches to try to figure out how to push such a direct current of 2 milliamp for up to 20 minutes through a small electrode. Many formulations did not work, but this is the one that did, and this is what we call high definition. So it's the electrode itself that's about 1 centimeter in diameter, Other aspects of the shape are critical. The use of sintered silver chloride over limited use is critical, and also the gel design is particularly critical. And that we make our own gel. The closest commercial gel we found that approximated the parameters that we need was Sigma gel. So that's already readily available. So doing this, you can stimulate with 2 milliamp through a small electrode with equal tolerability as with sponges. And high definition isn't just limited to the 4x1 approach, really high definition, and by that I really just mean using small electrodes to do tDCS. There's a tremendous amount of flexibility. So just as with EEG, you could theoretically have 64 electrodes on the head or 100 electrodes on the head, and you could independently control the current in each one. Now that leads to a tremendous amount of flexibility, right? So wait, if I have 64 electrodes and each one I can do any analog amount of current between -2 and 2, how do I do that? And so a significant amount of modeling effort has been devoted to targeting, and in 2011, we published a paper which suggested that given a target, so this is if you know the target, and you know also the orientation of the neurons in that target, you can in a single step produce a truly optimized solution. And this was because this problem ended up being a linear one. And that's why we could reduce it to a single solution without having the need to search or use some sort of genetic algorithms or machine learning to converge. But the thing is, you still need to know what your target is, right? And this is something Vince was just talking about that he used functional imaging for, right? So even if you assume that given an MRI, and by the way, you do need the MRI for this targeting, even if you assume that our models are perfect, and you find them 100% reliable, you still need to pick your target. And this is where, as Vince just introduced, this is where imaging comes in. And in addition to fMRI, it's natural to think about perhaps using EEG to guide stimulation, right? Oh, it also puts electrodes on the head, right? It looks a lot like, in many ways, like high definition tDCS. And the ideas of using EEG to guide transcranial electrical stimulation in general really go back decades, I think at least to the 70s. You can at least find patents, if not papers, of people talking about, well, why not use the EEG and I'll stimulate via the same electrodes? And it has things that seem to make it very appealing. One is that it's potentially easy. Well, I already got the cap on the head. I'm already gelling things up, right? So it's the same thing. It's no extra work. I'm just going to use some electrodes for stimulation, and I'm going to use some electrodes for recording, right? And I can, if I already have an EEG system, I can just replace some of the channels with stimulation ones. It's certainly individualized, even how much the cap stretches would theoretically be the same across subjects. There's a notion that this could be done automatically, but this is really the question then. How exactly are you going to use EEG, right? That's the real question. And if you come up with a solution that's very complicated, then it's certainly no longer easier. And then the notion of real time, and I'll say a few comments about that. So it's different to approach this problem offline as an EEG and then stimulation, and then online. So one way to approach it, sort of the straight way, I think, combining existing techniques, is you start with a very high resolution MRI of this individual, 1 millimeter in all access, just covering the whole head, which has its own costs and complexity. Then you conduct a super high density EEG on this person. Then you use the information from the EEG to localize sources, and people who work on this know how problematic this is. This is not a sort of a singularly constrained problem. There's many assumptions that go into it. And then with the MRI, you run a full high definition optimization to that target. And certainly this process is possible, but I don't think it's so easy anymore. And so the proposal was to go into scalp space and maybe do it instead like this. Conduct an EEG, perhaps of moderate resolution. Then at the level of the scalp, do what I'm calling a scalp space inversion. And what that means is that there's no scans needed, no assumptions about how many sources you have in the head and how they lead to these potentials, and some relatively easy math. And easy, I mean math that everyone can agree on is at least achieving what its goals are. And then conducting HD-tDCS with the minimum number of electrodes. And so this is the approach I'll be discussing, and this so far has been developed, again, in modeling. So this is always where we start with simulations. So what this is showing you here is, if you would imagine a single local dipole in the head that's sort of pointed along the surface of the head, we can simulate the potential that this will generate. So this is not stimulation. This is the dipole is inside the head. It's producing a very intense electric field inside the head, right? You can see that here at the, does my cursor come up? Yeah, there you see it right there. It's extremely focal. It's a small dipole. But on the surface of the head, you see something extremely diffuse, right? Because as that current moves towards the surface of the head, you can still kind of make out this sort of dipole shape, right? Positive voltages, negative voltages. So this is false color of voltage. And you can say, well, how could I start by inverting it? Well, one simple approach is to say, well, I'm going to take all the voltages that I recorded, and I'm just going to stimulate with them. So if I recorded 1 volt here, and 0.5 volts here, and negative 1 volt here, I'm simply going to apply those via the same electrodes. And by the way, this is being done with hundreds of electrodes in this stimulation. So we're simulating this. We are discretizing it, but we're taking hundreds of electrodes to do it. Well, when you do that, you do not reproduce. In fact, you do a rather bad job of hitting this target. And you could also say, well, what if I take all the voltages here, and I'll simply call them current. So where I have 1 volt, I'll apply 1 amp. And where I have 0.5 volt, I'll apply 0.5 amp. And when I have negative 1 volt, I'll apply negative amp. And this is the result that you see. And I should mention here that in all these cases, I'm plotting each plot to its relative amplitude. So red is just the highest intensity you'd see in that case. And I can come back to the absolute. This also, unfortunately, we found, but we expected, did not produce stimulation of just this tiny point. And it turns out that it's a really hard problem because single dipoles are always going to be really tiny. And no stimulation can produce stimulation this focal. And so maybe this wasn't a bit of a surprise, but clearly in principle, this approach wasn't even getting us close. And nor did we necessarily expect it to. We did another approach, and in this case what we did is we took each electrode and we calculated the Laplacian at it. We basically did the current source density at that electrode, which is commonly done with EEG, and we applied that current to each electrode. So we did the current density at each electrode, and then we applied that at the recording phase, and then we applied the stimulation at that same point. And this is the result of doing this with 256 channels. I'll show you less here. And you can see that certainly we're able to drive current across where we know the dipole is, because we put it there. It's certainly not particularly focal. And this is comparing using the Laplacian approach with just the voltage to current flip. I would say it's moderately better. And here you can see that what we're doing here is we're stripping away the number of electrodes we're using all the way down to 8. So starting with hundreds of electrodes and going down to 8 electrodes, something is lost. So 256 is great, but that's certainly cumbersome. It would mean you need not only a 256 channel recording system, you need a 256 channel stimulation system. And it's not like you're getting something particularly amazing as far as getting back to that target. And by the way, I should mention, I'm flying through this. I'm happy to address questions. There's some subtlety to how you do this. You have to do things like ignore. It's hard to do Laplacians around electrodes at the edges, so you would ignore them. You need to make sure that the amount of current injecting in at the anodes adds up to the amount of current coming out at the cathodes. But none of these things are particularly, once you realize them, they're not particularly things that would be debated at implementation. So what about going back to the most basic thing, right? The most basic thing you could possibly do and saying, well, I know I have a dipole here, and that's kind of producing this dipoling-looking scalp potential. What if I just took 2 electrodes, sticking to high definition, sticking to 2 small electrodes, and I put an anode here and a cathode here, sort of like where I see the positive and negative? What kind of current flow would that produce? And this is the result, right? And so if you've been paying attention so far, you see, well, that's not so bad, is it? It's an extremely simple approach. We could do it a little bit better than this, because this is sort of ad hoc, meaning we just kind of looked. A slightly better way to do it is you can actually do the Laplacian and then look across all the electrodes and find the single most positive and the single most negative and pick those, right, as where you put the electrode. So that makes the process automatic but not particularly complicated. And this is the result that we saw. You can quantify this in all kind of ways across all kind of heads, but you can see that the result isn't so bad. There's a couple things about this that well, everything's obvious in retrospect, right? But we're not obvious at the time. Because when we're doing this, we're kind of asking, well, what are we optimizing for? What did we just do here? So we knew from the same optimization paper in 2011, there's always trade-offs. If you want to be really targeted, particularly at the cortex, what you do is you bring the electrodes closer and closer and closer together. So if I have two electrodes here, right, you can imagine where the current's going to go. And if I put two electrodes here, it's not going to flow to my left ear. It's going to stay between the electrodes. And so it's obvious, but you can derive it mathematically, that the closer the electrodes are, the more focal you are. But the closer you are, the more of it flows through the skin and none of it gets into the brain. So it's a trade-off, right? You start to lose intensity, but you can keep going up with your focality. On the other hand, if you actually just wanted to optimize for intensity, meaning my goal is to get the maximum current into the dorsolateral prefrontal cortex, or M1, and I do not care what else in the brain I stimulate, the solution, according to the optimization, is once again a two electrodes solution, but the electrodes are across the head. And again, obvious in retrospect, right? But it took us a while to work this up mathematically. And where you put them, by the way, is not trivial, because it depends on the orientation. So if the dipole is oriented in a particular way, by dipole, if the cells that I'm interested in, the pyramidal cells in the cortex, are pointed in a particular way, you need to position the electrodes, the pair, in a very peculiar part on the head, right, to get that orientation. So depending on the orientation, I would come up with a very different optimal solution just based on intensity. So what did we do here? Honestly, we're still working on it, but I think what we did is we sort of hedged. So if you compare the best you can do with focality, you can do better than this, but you don't get much intensity. And if you want to look at the best we did in intensity, also you'll find that this is not quite right. But both those approaches, maximum optimization for intensity or focality based on the 2011 paper, requires an MRI. And requires you telling that optimization where the dipole is. Now, what if you don't know where the dipole is? What if you don't have the MRI? Well, this solution doesn't depend on those. I mean, here, yes, I'm running the model, so I needed the MRI to do the model, but to actually implement this, I wouldn't need that information. I would just need to do the EEG, Laplacian, maximum anode, maximum cathode, stimulate right there. And what's kind of pretty about this is we always get the orientation right. So we've done different controls, you can mess with the dipole and so on, but you get the orientation right on top of it, which is particularly important, we think, for how tDCS works. tDCS is considered to be very orientation dependent. And so the method in the very qualitative way, I've just introduced it today, and this is really the first introduction of it, is you collect an EEG. We do not think you necessarily need a super high density EEG for this purpose, and we can use models to figure out how dense you need to be, some simple math at all channels, and then two electrodes at the max and the min, with an outcome that we find exciting for now, which is you get the right direction, you get a nice balance between targeting and intensity, and most critically, it's simple. One permutation that I'm just going to say very briefly is, if the dipole is not sideways, but the dipole is pointed up, right, then the scalp potential ends up looking more like a unipolar distribution. And in this case, a slight variation is you would actually do the 4x1 high definition montage, which produces radial current flow under it. And so that's how you'd get back to your target. So there is one permutation where, depending on the nature of the voltage profile, whether it looks like a dipole or a unipole, you may either use 2 electrodes or 5. And 5 is not so much. Okay. I wanted to say a few things about concurrent, because obviously when we were doing this work, we were starting to run experimental pilots with several collaborators, and it's nice to go concurrent as opposed to sequential, right? I already have the cap on, right? Why don't I do it at the same time? We discovered quite a few problems that we're currently writing up, and I did want to flag them here. The first thing to realize is that no stimulation source is perfect. Even the ones made by the company I founded are not perfect. They have electronics, they have noise, and that noise, while very small for tDCS researchers, is huge for EEG researchers. And I think you can't say it's not there. because then you get into trouble. What you need to do is you need to quantify it, and once you know how much noise you have, then you can talk about whether you're really detecting real signal. Another problem we discovered was that stimulation alters the tissue conductivity. The most obvious thing that we know tDCS does is it makes the skin red. It changes blood flow in the skin, and as you can imagine, that changes skin properties, including temperature and its conductivity. Now, if you change skin conductivity, right, now you're changing, and it's not just skin, it's everything else, you're actually changing the EEG, because you're changing how the sources in the brain are then reflected in the scalp. And this is a particularly problematic artifact, because first of all, it's going to be localized to where you stimulate, because that's where you're going to produce the most skin changes, so it's montage specific. And it's time specific in a very problematic way as well, because the skin redness builds up, just like we think brain excitability does, over a few minutes. And it lasts after stimulation, so you're seeing post effects as well. And I need to emphasize, it's not just the skin itself. There's conductivity changes at the level of the whole brain. It's nice to think that the stimulation does not affect amplifiers. That has not been our experience. We used multiple stimulation systems across multiple high-end research amplifiers. It's not just the electrodes. It's not just the channels not saturating. Simply because they don't saturate does not mean that they've been moved into a regime where they're performing in a way that is just a little different because of the presence of volts on the scalp, for example, different filter settings, and then there are other issues like systems that use active things like driven right leg, and those have to be considered. Physiological artifacts, these are the most peculiar things we've seen, and I don't know what's causing them, but I know they're so obvious you can see them by eye. For example, eye blink artifacts are a big deal. In some cases with EEG, stimulation changes the eye blink artifact. It can actually invert the direction of it. I'm happy to hear theories as to why, but we saw retinal sensitivities also changed. We've seen things where the way the EKG is reflected in the EEG is being affected. And then other issues related to the electrode distortion when you try to stimulate and record from the same channel. All this does not mean that concurrent should not be done. It just means that it should be approached with extreme caution, just like concurrent anything, right? Concurrent EEG plus fMRI, concurrent tDCS plus fMRI. When you're combining two techniques, there's always issues with interaction. So that's really, would be my take-home messages. I think tDCS can be focal. It is true that when you do tDCS with two large 5 by 7 centimeter sponges across the head, it is not necessarily focal, but doesn't mean that tDCS is not focal. When you apply the same sophistication to electrical as people have applied to magnetic, you can make categorical steps forward as far as targeting. Certainly, though, you need to start being more aware of the kind of electrodes that you're using, but there's a formula. And as long as you follow the formula, we've had thousands of sessions, tens of thousands, I think, getting to this point. And the tolerability is comparable to regular tDCS. If you have an MRI of a subject, and you have some time on your hands, and you know exactly where your target is, because you can point to it, there are methods that exist today that allow you to design optimal stimulation parameters with any constraints that you want. I don't want to go over 2 milliamp total. I don't want to go more than 2 milliamp per channel. This is a pretty solved problem. But of course, you have to have the MRI and you have to know where your target is. And going back to Vince's point of making this something that's broadly deployable, we've recognized that even asking for a low-grade MRI will probably make this not deployed broadly in the clinic. Simply inverting the EEG to stimulation does not work, was not a huge surprise. We thought this might work. There's this thing called reciprocity. We thought we might be able to work in different ways, but we couldn't. But another approach, which was based simply in scalp space, using the Laplacian and picking just a few electrodes, either 2 or 5 worked. It's 2 if it looks like bipolar. It's 5 if it's unipolar. You may very well ask, what if it does not look like a bipolar or a unipolar, which it never will, of course. Where do we go from there? And I mean, I think people who work in EEG have different techniques they do use to pull these things apart in scalp space. And I think concurrent, so that would be recording, stimulating later, and so on. Concurrent is possible, but you need to be extremely vigilant, or things will be problematic. And I had one more idea slide, but how am I in time, Vince? Okay, so let me just throw out one more idea that, again, we have not published on yet. And this is an idea we're calling functional sets, which I think has application validity. The bottom line is if I take one HD electrode with 2 milliamp, and then instead I take 2, and I put 1 milliamp through each one, as far as the brain is concerned, they look the same. The brain can't tell that it's one source versus 2. There's too much blurring in between. So that means that I have 2 electrodes, and whether I put 2 milliamp in one and 0 on the other, or 1 on one and 1 on the other, 1.5 and 0.5, it doesn't matter. I can move things freely. And this opens up interesting dimensions as far as how we can further optimize technology. For example, if one electrode goes bad and we know it, we can shift current around. But it also allows us to decrease tolerability, right? So if 2 milliamp feels a certain amount, well, now we can split it into 1. And so this is an idea that we're currently developing, and it directly relates also to this inversion idea. So going here from 2 to 8, you can say, well, this doesn't look very different from here to here. Well, that could be a good thing. Right? That could be a good thing because with here we have 2 milliamp at the anode, and here, because there's 4 electrodes that are serving at the anode, I just have 0.5. Now the tolerability may drop to the point of borderline sensation, but certainly not painful. So we're enthusiastic for this idea, and it's all interrelated. So I want to acknowledge the people who did all this work. Really, there were several projects. One was related to the modeling, particularly Lucas Parra is a close colleague. The electrode design team were people who were stimulating their forearm for a very long time before we found the right formulation. Again, Franca, who's here, and her team in Italy, which basically did the EEG inversion and had the ideas to go to this simple approach. That was her student, Andrea. And then currently we're working on this issue of the problem with EEG and tES. Unfortunately, we don't have a lot of solutions, but we have a lot of bad data. And so we're going to be putting that into a paper to at least share with people the issues that we've discovered. So thank you for your attention.